Hello and welcome to 7 Minutes of Science. I'm your host, Eddie Edwards, here at Mission Acceptable, bringing you your weekly high-speed rundown of all the biggest stories in the world of science this week. In a week that brings us humans controlling mice DNA with their brains, deadly semen, and little story, don't know if you heard about it, but we landed a probe on a freaking comet. Cool. Anyway, so let's not beat around the bush, and without further ado, give me 7 minutes on the clock, and in 3, 2, 1, let's do this. Monday, in news that confirms a whole host of suspicions I've had for a very long time, researchers at Johns Hopkins Medical School and the University of Nebraska, led by Dr. Robert E. Yolkin, have found a virus that makes people more stupid. Yep, that's the picture I'm going with, stupid. And I do have a microphone, by the way, now. Cool, isn't it? Anyway, this all stems from a completely unrelated study dealing with throat microbes. During the course of that research, they found a microbe in the throats of some otherwise healthy people. Of the 90 people in the study, 40 of them turned out to have this microbe. Those 40 performed worse in activities designed to test attention span, speed, and accuracy of visual processing. Now, obviously, an important reality check is a very small test with very small people. However, even the notion that I might be able to catch stupid from people makes me very, very glad that the internet cannot transmit germs. Okay, then on to Tuesday's big story. As you may or may not be aware, human beings have been attempting to remove their hands from the whole using stuff thing for some time now. So we've had people using brainwaves to play games, move robots, and even move the hands of other people, if you remember last week's Sin Against Nature. Well, here's an insane use of the tech. Researchers have worked out a way to take a human brain up to mice DNA. Yep, how it works is the device scans the brain, looking for 3 distinct patterns like concentration, relaxation, things like that. This signal is then sent to a light implanted in the mouse, which then activates genes through optogenetics, which is when light is used to switch genes on and off. These genes make the mouse secrete certain proteins, which can then be picked up in the DNA. I wondered why anybody else would do this when I first heard about the story, and I still do. This only works with genes that have been engineered into the mouse ahead of birth, so this just seems like a really cool, drawn-out way of doing something that's not that worthwhile doing in the first place. And Wednesday. Wow, we got through those first 2 stories quickly, didn't we? Almost as if I'm squeezing all the other stories this week so we can have more time talking about one of them in particular. Wonder why that could be. We landed a probe on a freaking comet. I'm truly glad to have started the show when we did, as covering this has been a genuine pleasure. Fans of the show may be aware that over the past few months, the ESA's Rosetta mission has been moving closer and closer to its cometary play, Comet 67P. Well, Wednesday saw the fruition of a decade of scientific work. So what happened? At 0630 GMT, which by the way is Space's time zone, the probe performed its pre-delivery manoeuvre, turning to face the comet, putting it in a position to release the Philae lander. I almost forgot to mention that the times I'm giving here are Earth confirmation times. It takes 28 minutes for a signal to get there and 28 for it to get it back. So every time they're doing anything on this, there's an hour of wait time, which is incredible and must have been an incredibly tense hour. So yeah, they doing that. The probe then is fired. Basically the probe then was in position. 7 o'clock. The team made their fourth no-go decision after the probe confirmed it successfully changed position. In 1903, the probe used 3 rotating screws to unhook and nudge the lander towards the comet. Between 9:04 and 9:47, the lander fires up its equipment, snaps a picture of Rosetta, and swivels its body 14 degrees relative to the legs to get in a good landing position. At 19:04, the lander was clear of the probe and performs another maneuver to push itself back into stable orbit so it can relay the lander's data back to Earth. At 10:53, the probe and the lander successfully connect, and the lander starts uploading info, including the picture taken of the probe. 14:58 to 15:17, the lander fires up its active defense system, basically a stability thrusters, and the harpoons it has in its legs to anchor itself to the comet if needed. At this stage, it also starts using the descent camera to capture some thoroughly unique pictures. At 16:03, we, the human race, had landed a probe on a comet. Now, some later reports stated that the harpoons failed to launch, and as such, the lander did bounce and isn't secured enough to start gathering samples and data. But with everything in position and the team that managed to pull this off in the first place working on a solution, I'm confident they'll manage to find one. Also, as a Brit, I've got to say, I love the fact that we landed on a comet, because we were rebuilding from World War II while Russia and America argued over the moon. So it's nice to have a big landmark fall squarely in the European realm of perfection. There's too much of this to really talk about. I'm going to upload 2 other videos, one going into more detail about some of the problems that are being experienced, and another one that's going to go through some of the pictures taken by the probe as it's been on its journey to kind of form a narrated timeline, because I cannot do this justice in the time allotted. Right. On to Thursday, chemists Alan Jacobson, Onjin Middlejak, and Olaf Douglas, working at the University of Houston, have created a molecule that binds with greenhouse gases. This isn't exactly news, as this has been achieved before, but these guys have improved upon that work The previous attempts to create something like this, of this nature, have usually run into the problem that the molecule is too heavy, as they've had to use materials in their construction, like metal. This new solution uses individual molecules which are only bonded by weak interaction, which means that the whole structure is stable in water, incredibly lightweight, and can be broken down into its constituent parts, relatively easy for retrieval. This won't be of any use against carbon dioxide, but it does work on several other greenhouse gases, which are less common but significantly more harmful to the environment, like fluorocarbons and freon. Friday, your sperm is trying to kill you, or unless of course you're a lady, in which case my sperm is trying to kill you. To be fair, that started out as a bit of an attention-grabbing joke, and now I've realised it's gone horribly wrong, so I'm just going to reject from that and go. Anyway, I'm fairly sure that over the last few years you've heard there have been many stories about breakthroughs in dealing with and treating HIV. Yet, here we are, still sharing a planet with HIV. What's up with that? Well, it turns out that when you treat this in human trials, and they've done this now in a lab with in real semen and real vaginal fluid, and it turns out that semen actually kills the antimicrobial stuff that they're using to kill the HIV. Makes sense if you think about it. Nothing's going to come microbe is actually going to be that nice to sperm, so it makes sense in a protection. From a protection point of view, that it would do this. However, the good news is one of the treatments, maraviric, which targeted the receptors in the viruses, the receptors in cells that the viruses use to get inside, turns out to be still as effective, very effective. So it looks like that could be the solution to HIV. Very useful information. And finally, we have some worthwhile scientific data in the study of the effects of marijuana on the human brain. I don't care where you land on the cannabis debate, but trust me, you don't really land on the side of scientific data because there isn't a lot of scientific data. You see, both sides of the argument want to claim either it's going to kill you or it's going to be a miracle cure. For years, any research has been kind of shaky because people don't want to admit that they're doing something illegal and get arrested. But some places that legalised it have legalised it for so long that they can't really see the point in doing this kind of studies. Other places haven't. Anyway, somebody has now looked at the human brain in long-term users of cannabis with 3 different types of brain scan, and it has revealed 4 things. 1, pot smokers have smaller orbital frontal cortex. 2, friends of Mary Jane also increase brain activity. Playing on the grass will lower your IQ and the total amount of matter in your brain, and the amount of damage that green will do is linked to how young you are when you start, which seems to me to indicate that the best policy would be a policy that regulated this and kept it out of the hands of children. Okay, and finally, in not really science but sort of news, some of you may remember a few years back 7 scientists were arrested, charged and sentenced 6 years in jail for failing to predict an earthquake in the town of L'Aquila that killed 309 people. This was done because everyone who lives in Italy is either a moron or too stupid to win an argument with a moron. Well, in a bit of good news, a judge by the name of Franca Navadra, which is Italian for smarter than inanimate object, has overturned the ruling. So well done for Italy for finally being intelligent again. You can't lock up your scientists for stuff like that, and we are done. Boom. I was very concerned I wasn't going to be able to do it this week because that middle story about the comet is a really huge thing. That's why we're doing the 2 extra videos. Links will be here. I'm pointing with both fingers. So one will be, like I say, a narrated timeline of some of the photos that we have from the Rosetta probe, just going through how the journey, because this thing has been travelling now for a decade it's taken to get to its location, and I can't do that justice in this thing, but I do really want to talk about it with you. The other thing we're going to do is run through some of the problems and snags that have hit. I do record this on Friday, and there was some information coming through late Thursday that where the probes landed, it might not be getting power to the solar panels. So what I'm actually going to do is, before I upload this on Sunday, I'm going to record another 4 or 5 minute video, just going into the details about the problems that have been had with the probe, what sort of solutions there are, what this means for the project. Because again, I could do 7 minutes entirely on this show. I could do 7 hours on the Rosetta mission. It's incredibly cool thing that we're doing as a species. So, anyway, thanks for watching as always. As usual, if you like the video, like the video. If you think somebody you know would like the video, share the video. And if you want to see more of what we're doing here, do subscribe. We'll be having new commentary videos coming up, which is me commentating, doing sports commentary to police videos, and we've got some other sorts of material we're going to be looking at on that front. And we've got a couple of our Let's Plays and our gaming videos will be going up live this week. Some scheduling problems and some issues with tech, but everything's up and running now, so hopefully that'll become a regular thing that we do. If you want to see all of this stuff, this great and exciting stuff that we are doing here, please subscribe to the channel. And as always, good luck in your endeavours. If your endeavours be good. Goodbye. Over 10 years ago.